Hey y'all, as promised, here is the quick lecture overview with some detail on chapter 10, meiosis and sexual life cycles. Okay, so here is a picture of three people. I chose this picture because it is my cute little baby nephew. And as you can see here, he kind of looks like the lady who is his mom, and he kind of looks like the guy who is his dad. And that is because of meiosis. Okay, so that's what we're going to talk about, sex. Bow, chicka, bow, bow. So when you think meiosis, you should be thinking making gametes or sex cells. Okay, so gametes are eggs and sperm. All right, let's take a quick look at what the key concepts are for this chapter. You should know the purpose and the definition of sexual reproduction. What is sexual reproduction? Okay, you have male, female, bow, chicka, bow, bow. They fuse and you get a zygote, okay? We are also talking about reducing the amount of genetic material when we make gametes, okay? So you have learned thus far, and in your previous module, you have learned that you have 46 chromosomes in all of your cells, okay? That is with one exception, your gametes, okay? Eggs and sperm have 23 chromosomes, right? So that when they fuse, when sperm meets egg, then you go back to 46 and you go back to doing mitosis. Okay, so understand what it means when we say alternation of haploid, diploid, or diploid. Diploid is 2N, right? That is your 46, your somatic, your all of your somatic cells. So all the cells in your body have 46 chromosomes, whereas the sperm and egg have 23. Okay, so that is the haploid state. I actually cannot remember if I put that slide in here, but also realize that in your textbook, it describes this, that not all organisms do this the same, right? The chapter is entitled Sexual Life Cycles, Meiosis and Sexual Life Cycles. So plants and algae do this differently than we do, and so do fungi. There are different types of sexual life cycles, okay? What you really need to know in order to succeed in this module is what happens in each phase of meiosis, and we're going to go through that. In your worksheet that you have for this module, there are chromosome diagrams. Can you draw those? Okay, can you identify the different phases? Some of you have been emailing and asking questions and doing really well in understanding the different phases of meiosis. And then since you learned about mitosis in the last chapter, can you compare and contrast them? Do you know the difference? Okay, and hopefully by the end of this, you should have a really, really good idea. Okay, here are some keywords here that are really important in giving us some context of what we're learning about now. We're learning about genetics, okay? The study of heredity and variation. So heredity is the reason why you kind of look like mom and maybe you kind of look like dad, right? I have my dad's eyebrows. Thanks, Pops, right? And my dad's nose. Thank you for that. I inherited those traits from my dad through fusion of sperm and egg. Gross, right? And then there's variation. So I have two siblings. I have a sister and a brother, and they look similar, but not exactly the same, right? If you think about your family, look at your family. Who do you look more like, right? There's some kind of commonality between all of you, and there is a lot of variation, and this variation is really, really, really important when we talk about evolution and natural selection. It is the substrate that those mechanisms work on, right? So if you remember way, way back in the beginning of the semester, we talked about natural selection, and we looked at the example from the textbook with the beetles, and the beetles, I think they were beetles, they had all different colors. And some of them were more susceptible to being spotted by a predator than others because of the variation between or within that population. Okay, so variation is really important when we talk about natural selection, and we're going to look at how we get that variation out of meiosis. Hopefully, before I say it explicitly, and hopefully you have already engaged with the material, you'll realize that the products of meiosis result in genetically distinct cells, as opposed to in mitosis, we're making genetically identical daughter cells. Okay, so there's one big major difference between meiosis and mitosis. Okay, so here they are. Okay, so for the sake of simplicity, again, we're using chromosome diagrams. Okay, we're not going to draw all 46 chromosomes. Here, our organism has a total number of 6. 2n number equals 6. So you have your egg and your sperm are each going to contribute half of the chromosomes in your offspring, right? So maternal set has 3 and the paternal set has 3. Half from mom, half from dad. Okay? What you see here is your whole set of chromosomes, right? They're duplicated chromosomes, so they have sister chromatids, right? But this is still one chromosome, and you got a maternal set and a paternal set, okay? Diploid, haploid, okay? This is the diploid, and then your haploid would be here. These are your haploid cells, your sex cells, okay? And then they fuse, and now you have the full genetic makeup, okay? Right, again, we're focusing mostly on humans, although you have to realize that other organisms do it differently. And we're not going to focus too much on that because corona. Okay, so we do it similarly as all animals. We are multicellular diploid organisms, right? We have many, many cells that make up our body, and we have our diploid number is 46. We do meiosis to make gametes, which have a haploid number, and the haploid number, half, is 23. You need a male and a female, or you need gametes from males and females, right? We have so many different ways of doing this nowadays. And you basically just need a sperm and an egg, doesn't matter where you get it from or how you get there, and you fuse them, and now you have your diploid zygote with the full complement of 46 chromosomes, right? And then in order to go from a zygote to growth and development, the reason for mitosis, you do mitosis, okay? And here it is. Yeah, I am mentioning it. I don't want to go too much into detail with it, but we do have some organisms that actually alternate between being a diploid multicellular organism and a haploid multicellular organism, right? So some plants make gametophytes, and that is a multicellular organism with a haploid number, right? And it grows into a multicellular organism with half the amount of genetic information. And then fungi and protists also do this differently. We are going to focus on animals, okay, and talk about meiosis. So again, the goal of meiosis is to produce gametes or spores in some organisms, and you are making haploid cells. Okay, if you keep this in mind, you will be able to differentiate between mitosis and meiosis, and you'll be able to have a clear picture in your mind. Okay, so mitosis, we're making genetically identical cells. 3 reasons why we do mitosis. If you are a bacterium or a organism like the hydra that is described in your textbook, right, you may be doing mitosis for asexual reproduction, right? We also do mitosis for this to happen, right? Growth and development, going from zygote to multicellular organism. And then the third thing was you get a boo-boo, right? Tissue repair, tissue damage repair. So wear and tear, normal wear and tear, and then damage. You need mitosis to repair that tissue. Okay, so mitosis has its reasons. Meiosis, the only reason is to make haploid gametes. Okay, you want to reduce the amount of chromosomes by half. All right, so let's look at an overview of what meiosis is. And now you should already be familiar with some of these key terms. Okay, so in this diagram, we're looking at, right here, we're looking at a homologous pair of chromosomes in a diploid parent cell. What does that mean? That means it has all of the genetic information. This could be 46 chromosomes. You got half from mom, half from dad. Okay, you undergo, in order to get ready, you're in interphase, right? The cell is going to get ready to undergo meiosis. So what it does is it's going to duplicate the chromosomes. And now you have sister chromatids. We're going to get ready. It's the S phase. So you have one round of DNA replication, and then you have 2 rounds of cell division. And what that should tell you is that you end up with not 2 daughter cells, but 3, 4, where'd my 3 go? 4 haploid cells with single chromosomes, right? Because we duplicate it and then eventually we split the sister chromatids. Okay, so in meiosis 1, what you do is you take your duplicated chromosomes and you separate your homologous pairs. Okay, so let's say we're talking about chromosome 1. You have one from mom, one from dad. In the first round of meiosis, you separate them into 2 separate cells, but they are still duplicated. They still have sister chromatids. And then in meiosis 2 is basically like mitosis in that you take the sister chromatids and you separate them into 2 separate cells. So look, you started with 2 cells here, and then each of those splits into 2, okay? And then what you end up with is a haploid cell. So if you look at the parent cell, you had a pair of chromosomes, whereas if you look at the parent cell and then in the haploid cell, at the end of meiosis, you end up with half the amount or 1 chromosome, okay? So meiosis only takes place in cells that are destined to be gametes. They're called germline cells, okay? And also DNA replication, this should have been clear, and if it wasn't, it should be clear now. When we replicate DNA in the S phase of interphase, we're not changing the number of chromosomes, right? They're either duplicated chromosomes or unduplicated chromosomes, right? If they're duplicated, they have sister chromatids. If they're not duplicated, they're just chromosomes. They're condensed chromosomes. Okay, so we're not changing the number of chromosomes when we make sister chromatids. Okay, I hope that's clear. Okay, those are your words. You can look at them at home. I'm just going to leave them there. And let's go through the phases of meiosis. Meiosis, making gametes. Okay, so in prophase 1, again, we're going to do P-mat. Okay, we don't really look at pro-metaphase in meiosis. We just, because in that chapter we were looking at a little bit more detail. We looked at chromosomes condensing and then the nuclear envelope fragmenting in pro-metaphase. Now we're just going straight to prophase 1. Still the same sequence of events. We're just not adding that extra phase in there. So P-mat. Okay, and we have 2 rounds of it. So we're going to have prophase 1, In prophase 1, this is really like your book talks about it as the busiest time for the cell. This is where all of the magic happens, okay? Things begin to happen. So the nuclear envelope fragments, the DNA is condensed, and what happens is, yes, you have your mitotic spindle, right? Your spindle microtubules form. And the really, really important thing that's happening here is the homologous chromosomes pair, okay? So your one from mom and one from dad are going to line up next to each other, and then they're going to cross over this thing called the chiasmata, which is basically an X, is going to form. Right, these guys are what's called in synapsis, and you actually exchange genetic information. You have genes, DNA that encodes for particular genes moving from one homologous pair, from one of the components of the homologous pair to the other one. Okay, so pair, one from mom, one from dad, they come next to each other, cross over, and exchange genetic information. Okay, that is really important. And that's what contributes to genetic variation. Okay? Metaphase, you should know. Metaphase starts with an M. Things are at the middle. Okay? So now the homologous pairs are lining up. And all you're doing in metaphase 1 is you're going to separate the homologous pairs. So you still have sister chromatids. Okay, you got your spindle fibers, your kinetochore microtubules attached to the kinetochore of the chromosome in the centromere, and they attach one to one of the homologues, it's been a long weekend, and the other side to the other homologue. Okay, so that means that each chromosome is attached to one side, so that when anaphase happens, anaphase 1, you are now separating homologues in that each of the homologues is going to end up in a separate cell. Okay? What is important here? This is very, very different from mitosis. We did not separate our sister chromatids. Our sister chromatids remain attached at the centromere, okay? And now you still have sister chromatids attached, but what you've done is you have halved the genetic information, okay? Now, we'll get to this, but independent assortment, keep in mind, of maternal and paternal chromosomes is happening here, right? So if we go back to our metaphase plate, you can see you have the red, I think, is from mom and blue is from dad, right? These lined up in a particular way. You could easily line them up in many different orientations, right? So you have red, red, blue, then blue, blue, red. What if you switched these and you ended up with all the red ones on one side and all the blue ones on the other side? You would have, the result of that would be a completely different genetic makeup of your daughter cells. Okay, so that is what independent assortment is, and we'll talk about it a little more towards the end. So anaphase 1, pulling apart your homologues. Okay, they remain duplicated. You end up in telophase 1, you remake or reassemble your nuclear envelope. And now look what happens. If you look at these chromatids, these sister chromatids, they're no longer identical because you crossed over and exchanged information between your homologous pairs. Okay, so telophase 1 followed by cytokinesis, you end up with two genetically distinct daughter cells with duplicated chromosomes. Okay, now what you're going to do in meiosis 2 Same thing you do in mitosis. You take your sister chromatids and you separate them. Okay, so you have, this is followed by interphase, right? But in this interphase, in between meiosis 1 and 2, you're going to not allow DNA replication to happen because you already did that. And after a certain amount of time, you're going to again divide these cells. Okay, so meiosis 2 is basically very similar to mitosis, except we are talking about meiosis, which means we are making gametes and we are producing genetically distinct daughter cells. Okay, so meiosis 2 begins when haploid, when you get, it begins with a haploid cell, half the amount of genetic information, whereas with mitosis, we started with all 46 chromosomes. Okay, so what happens is same thing, P, mat, okay, and now we're in the 2. And a couple of things to keep in mind. In prophase 2, we don't have as much busy work as we did in prophase 1. Sorry, y'all. In metaphase 2, we are doing the same thing we did in mitosis in that we are separating sister chromatids. Okay, anaphase 2 is where they come apart, and then in telophase 2, you end up with 4 haploid cells. Okay, and each one is genetically distinct from the next. Okay, you're not making this genetically you are not making genetically identical cells. So one thing you really should be aware of at the end of this lesson, you need to know the differences between meiosis versus mitosis. And if you think about the big picture, you will be able to tell me what the differences are. Big picture in mitosis is what? What is the result of mitosis? You can think about the result, and you can think about the result and the reasons. Okay, so the result, genetically identical cells. The reasons, asexual reproduction, tissue damage and repair, growth and development. Meiosis, one reason, we want to make gametes. Okay, in order to make gametes, you want to half your genetic information. Okay, go from 46 to 23 chromosomes. You also want to make sure that they're each genetically distinct. Okay, so that you have synapsis and crossing over. That's what that means when you have the homologous pairs coming close to each other in prophase 1 and crossing over, exchanging genetic information. Okay, in meiosis 1, we don't separate our sister chromatids. Okay, we just take half of my homologous pair in this cell and half of my homologous pair in that cell. Okay, and then this is a technicality, right? How does it molecularly happen? Those kinetochores of the sister chromatids that attach to the microtubule spindle in meiosis 1, one is going to attach to this side and the other one is going to attach to this side, so that one chromosome goes in this cell and the other chromosome goes in this cell. Whereas in mitosis, you have your duplicated chromosome, right? You have your centromere, you have your kinetochore microtubules. One is going to attach to this side and the other one is going to attach to this side so that you can pull apart your sister chromatids. Okay, and then one thing to note here is that DNA replication is going to be suppressed in that interphase between meiosis 1 and meiosis 2. Okay, so the goal of meiosis 1 is separating the homologs, right? You end up with half the amount of chromosomes in your resultant daughter cells. And then of meiosis 2, you have half the amount of chromosomes, but they're still duplicated chromosomes. So because they're duplicated chromosomes, you want to separate the sister chromatids. And that's basically what you do in mitosis. Okay, except in meiosis, we have genetically distinct daughter cells. Okay, when we make eggs, it's slightly different than when we make sperm. I think Hank in Crash Course talks about how when you make sperm, your 4 resulting sperm cells are all viable sperm. Whereas when we make eggs, only one of them is viable because that one takes most of the cytoplasmic resources in order to be a viable cell. The other 3 are just called polar bodies. I will not ask you about that. Just be aware that when we make sperm, it's slightly different than when we make eggs. Okay? All right. Really important concept to understand after we talk about meiosis, after we talk about making gametes, because now we are producing genetically distinct organisms, genetically distinct gametes, and then those gametes are going to fuse to produce genetically distinct organisms, you and your siblings. Okay, all of the genetic information, all of the genetic variability that we have worldwide. This is funny because it's quite relevant right now. We have this virus that is out to get the human race, right? It is no way in hell going to wipe out the entire human population because of the amount of genetic variability that we have within our population. Okay, so there are 3 mechanisms having to do with sexual reproduction that contribute to genetic variation. The first one is independent assortment of chromosomes at the metaphase plate, right? How do you line up your homologous pairs? Sorry, this is really bad drawing. Okay, I'm trying. Okay, but if you switch back a few slides, I will make you dizzy real quick. If you go right here, okay, there are many ways that you can organize these at the metaphase plate, and that is what's called independent assortment or independent orientation. You hear it being called both ways. So that contributes to genetic variation. Okay, crossing over, right? It's not just that you're taking one from mom and one from dad, okay, because then you would look a lot more similar to your siblings. You also have crossing over of genetic information between the homologous pairs, okay? And then even after you make gametes, that one sperm is going to randomly fertilize that one egg. There are many possibilities that contribute to the resulting organism. Okay, so this is really important in that you have genetic variation within a population, and that makes us less susceptible to disease as a human population. Okay, so we are increasing genetic diversity by doing sexual reproduction as a result of meiosis, right? So if you think about the number of different combinations that you could have at the metaphase plate, the way you would calculate that is you would raise that to the power of the number of haploid chromosomes that you have, right? So 2 to the n in our case would be 2 to the 23, and that would be a very big number. Okay, and then if you factor in the fact that one sperm is going to randomly fertilize one egg, you end up with something on the order, I can't remember the exact number, but it is on the order of trillions of possibilities for the genetic makeup of your resulting organism. So many opportunities. Okay, and this is just to show you 2 probable arrangements for organizing chromosomes at the metaphase plate in meiosis 1, right? You could organize them like this, or you could organize them like that, and it will give you 2 distinct different possibilities, right? And then you separate the homologs in metaphase 2, and then your daughter cells will end up with 4 different combinations there. Okay. All right. that is basically chapter 10 in a nutshell. Okay, 2 questions. These are from your textbook. So go to the back of the textbook, do your practice questions, finish your worksheet, do your quiz, and we are moving on to Mendel and the gene idea. I will be posting a lecture for that. I will try and get some practice problems up for you guys where I sit and do practice problems to get y'all ready for lecture exam number 3. Next week, you will have from Monday until Wednesday to get it done, and it will only cover 3 chapters, so no big deal, okay? Chapter 9, Chapter 10, and Chapter 11, okay? And then we will get what we can going into the final, right? That exam will be on Canvas. Okay, if you have questions, email me, holler. I will have office hours tomorrow. I can't remember what they are, but they are posted on your homepage. Bye, be well.